Our next speaker is on a similar theme, the interaction between the clinic and the lab, and Professor Tony Kelleher from the Kirby Institute is going to talk to us. Thanks, Tony.
Thanks very much. Unfortunately, I don't have a quote from Jello to go with that one. Just my potential conflicts of interest. And what I'd like to cover are two stories around, one around immune evasion and neutralizing antibodies, and demonstrate to you that convalescence is not passive and that there is obvious evolution of the immune response in population-based studies. And then I'd like to touch on ongoing inflammation post-COVID and look at the complications of convalescence and immune activation. So in terms of neutralizing antibodies and viral tropism, we've developed in the lab, mainly around Stuart Turville, a suite of multiple assays that allow us the rapid isolation from swabs of the virus, and then its characterization phenotypically, both in terms of its ability to evade the immune system and also its ability to infect cells. This has been developed using human cells rather than VOE6s, and those cells have been transfected with levels of with ACE2 and TMPRSS2. And we actually have a suite of these cells that allow us to isolate rapidly isolate all the variants of concern. This is married with high throughput sequencing of the sort that Bill just mentioned. And in fact, this has been a close collaboration with Bill and also with Dominic throughout. And we're able to rapidly generate genotype-phenotype correlations. As important as all those very elegant assays are, they're nothing without really good clinical data. And we've had excellent access to cohort samples, cohort and biobanks collected systematically, such as ADAPT, which is a convalescent cohort, VIM, which is a vaccine cohort, and then access to hyperimmune globulin and IVIG produced by CSL Behring. So neutralizing antibodies and pre-Omicron variants. So as what you can see here is that whether you develop the monoclonals, the immune neutralizing response from post-vaccine cohort on the left or post-infection cohort on the right, and whether you look at individuals or whether you looked at pooled immunoglobulin IgG from hyperimmune preparations developed from post-vaccine or convalescent donors, that most of the variants of concern pre-Omicron were relatively non-immunoevasive. And in fact, the most immunoevasive of those variants of concern was beta, which was about 3 to 5-fold, reduced neutralization capacity by about 3 to 5-fold relative to the ancestral variants. And in fact, Delta had very little immunoevasive properties at all. This is in contrast with looking at the same sort of data with Omicron, where this increased immune evasion by between 1.5 to 1.5 logs, depending on how you consider the extent of immune evasion and the source. So the 4 graphs show the rate of neutralization in various cohorts, either convalescent cohorts infected with various strains of the virus, or with indeed the people with hybrid immunity. And you can see that there is a decrease in neutralization capacity between 0.5 and 1.5 logs. And this is true again with pooled immunoglobulins, and you get again a 5 to 10-fold decrease in the ability to neutralize the initial Omicron variants. So to remind you, Omicron had 37 mutations in spike when it was compared to Wuhan. 15 of these were in the N-terminal domain, and 11 of those were in the receptor binding domain, and it bound ACE2 with greater affinity. And some of the examples in variants are shown there on the right, and you can see that they cluster. And what's happened is that those mutations have been sorted and resorted across the variants that have become, that have developed over time since the advent of Omicron. And these have divided into various families that we all know, but no one of these variants has become dominant. And in fact, at the moment, it's the B 2.1 strain and the BF strains that are dominant but even these represent a minority of the strains that are circulating, and there is an essential soup of these strains in the Australian population at the moment, and these are the data dating backwards from about the middle of January this year. So one of the ways that we've been able to look at this is we've had access to Privigen, which is Privigen batches produced by CSL Behring in the United States. So every about 3 to 4 weeks, CSL make a new batch of polyclonal purified IgG. And each of these batches represent the results of about, derived from about 10,000 separate donors. In these slides, the Delta wave is shown in grey, the BA1 is shown in pink, and the BA2 wave is shown in blue. In the bottom slide there, the black bars show the rate of uptake of vaccine in the American population, and the grey bars show the number of infections, with the increase in infections correlating with the Omicron wave in December. The red line shows the presence of nuclear capsid antibodies in the IVIG preparations, which follow the uptick in the rate of infection. Across the top of that, in the top graph, it shows the black line shows the neutralization capacity within these batches to the Wuhan strain, and then in the pink, the Delta. And then the other lines are the various variants shown there from BA1 through to XBF of the Omicron lineage. And you can see that, as I've shown you before, there is a significant hit taken to the neutralization capacity, but all the Omicron variants really within a relatively narrow band of each other. And if you look at this in a different way, if you look at this in breadth, you can see that the breadth, these IVIG preparations have been able to uniformly cover Delta to the same rate throughout this. There has been an increase in the ability of these pools to cover the Omicron strains, and in fact, the ability to cover the Omicron strains actually precede the Omicron strain occurring in the community and represent cross-reactivity from the vaccines initially and then from the other Omicron strains. So you're actually getting immunity in the community before the strains actually present themselves. If you look in a different way, these are now from the vaccine and convalescent cohorts showing people who have had 3 doses plus infection, 4 doses of vaccine, or 3 doses alone, or 3 doses following infection. And what you can see is, as I've shown you before, there is a significant hit taken to the neutralization capacity by the Omicron strains. But across all those variants, there is not that much difference between the various Omicron strains. And the difference between the worst and best of the Omicron strains is around about 4-fold and often less. And you can see that if you've had 4 doses of vaccine, you take a lot less hit across these variants. At the same time, we've looked at the ability of the infectivity and the entry characteristics of each of these viruses. And one of the insights I think we've got is from this graph shown as A, where we've compared the CT value in the swab against the TCID50 that has grown directly from that swab. And you can see that Delta produces far more infective viral particles per CT value than any of the other Omicron variants. And in fact, the BA.5 variant is better at producing virus than the subsequent variants. And that is then again summarized in E with the infectivity to particle ratio. Also, we've demonstrated that Delta, which is no surprise, infects alveolar cells better than BA.5, which is better at infecting alveolar cells than BA.2. And the TMPRSS2 usage is strongest with Delta and far less with Omicron. But the variation in the ability to use TMPRSS2 is not that much different across all the Omicron variants. So what I'd put to you is that Omicron recent strains have evolved a range of different mutations in RBD and the N-terminal domain. They have similar levels of immune evasion and viral infectivity. And I think this is looking like convergent evolution. I'm going to put a positive spin on this and say that Omicron appears to have reached a plateau of immune evasion while being able to maintain its transmissibility and infectivity. And it doesn't seem to be able to take the next leap. Vaccine boosting with the ancestral strain effectively increases neutralizing titer and breadth in individuals across populations to strains that the populations have not yet seen. And I think the rapid reproducible genotype-phenotype correlations that I've shown you here are really important, understanding the correlates of risk going forward. I'd also just add that XBB 1.5, which we've taken a while to isolate, shows very similar characteristics to its parents, XBB1. I just want to switch gears quite a bit now and now look at convalescence and long COVID. So this is the ADAPT cohort, which was initially the first 105 patients recruited through St. Vincent's run testing sites in the community in Sydney. And so this was a community-based cohort. Only about 15 of these patients were admitted and only 3 of them ended up in intensive care. At 4 and 8 months post-convalescence, the most post-infection, the most common symptoms that continued were fatigue and shortness of breath. And 30% of people in this cohort had symptoms that were consistent with long COVID at 4 months, and these persisted out to 8 months. So we took those 31 long COVID patients and matched them with 31 age and sex matched controls from the same population. Importantly, using the multiplex approach that Bill mentioned earlier, we had 25 patients recruited simultaneously from these community testing sites who were diagnosed with one of the other 4 prevalent coronaviruses, and we recruited them to the same follow-up. And I think this is a really important control. And then we had 16 healthy controls from early 2019. They were examined at 4, 8, and 24 months post-diagnosis. Remember, these are first wave samples. They were all unvaccinated, and they were all infected with the ancestral strain. So the difference between viral loads between the long COVID and matched controls at diagnosis were not different. And we measured 31 serum analytes. And what we found was that there was increase of 6 cytokines: interferon beta, interferon lambda, CXCL10 or MIG1, CXCL9 or MIG1, CXCL10 or IP10, IL-8 and soluble TIM3. And these were raised equally at 4 months in both the long COVIDs and matched controls. So a primary infection with this virus in an unvaccinated population causes prolonged immune dysregulation. By 8 months, most of the cytokines had returned to normal, but interferon beta, type 1 interferon and interferon lambda a type 3 interferon, was still raised in the long COVIDs, but were not raised in the matched controls. And as I said, all the others had returned to baseline at this stage. When we took all the data and looked at it at 8 months using a log linear model of classification, we found 4 analytes predicted long COVID with 81% accuracy, and these were interferon beta, pentraxin 3, interferon lambda, and IL-6. So what happened post 8 months? So we've followed these people out to 2 years. We had good retention. 59 of the 62 turned up for their 24-month visit. 59 of the 62 had at least 2 vaccinations, but only 45 of the 62 had 3. Now, in terms of interpreting the data, it's important to note that the 24-month visit happened on average about 3 months after the 3rd vaccination. The long COVID symptoms had resolved in 10 of the 28 at 24 months, but 3 of 31 of the matched controls had developed long COVID symptoms, and 2 of these appeared to be related to COVID reinfection. And during this period of follow-up, 4 of the 62 patients had documented COVID-19 reinfection. So by 24 months, of those cytokines that we saw raised at 4 months, only interferon beta remained raised, and this was in fewer of the patients, but still was significantly different. We had also measured a bunch of other things via flow cytometry, and we'd found at 4 and 8 months that there was depletion of a range of lymphocyte subsets, particularly a loss of subsets of naive T and B cells, a loss of subsets of certain CD8 effector cells, and at 8 months we'd seen no reconstitution of these naive T and B cell subsets, suggesting ongoing immune activation. But by 24 months, these had normalized between the 2 groups. We'd also, consistent with that innate sort of profile from the cytokine studies, we'd noted that there were activated monocytes and activated PDCs, plasmacytoid dendritic cells, at 3 and 8 months, but we had not seen activation of monocytoid dendritic cells. By 24 months, these had resolved. So convalescence from primary COVID infection in the absence of vaccination is characterized by prolonged immune dysregulation, regardless of the symptoms in an unvaccinated cohort. Long COVID has a diffuse immunological footprint characterized by increased type 1 and type 3 interferons, increased activation of plasmacytoid dendritic cells and monocytes, depletion of certain subsets of CD4 and CD8 naive T cells. I didn't show the data, but there's increased expression of PD1 and TIM3 exhaustion markers on CD8 T cells. And there also seems to be a correlation between the levels of the interferons and CNS symptomatology. Again, I didn't have time to show you that data. What's the cause of this? Well, we don't know. There may be prolonged antigen persistence, but I have to say that studying the peripheral blood does not back this up. There's as much antigen persistence in those with long COVID as in those without long COVID. It may be related to the autoimmune phenomena that others have described, or it may be cycles of epithelial and endothelial damage and repair. At the moment, we're trying to collate these data against a bunch of studies that were done on these patients, including COG state, the VAS fatigue scale, and the MRC dyspnea score, to look to see if there's any correlation with the cytokine patterns. And we're reassessing the pattern of dysregulation in those post-Delta and post-Omicron, and also trying to account for the effect of vaccination in those populations. And we're looking at tissue studies for antigen persistence, particularly in plasmacytoid dendritic cells. I'd just like to acknowledge the people who did the work, because I did none of it. Chan Fetsafan led the laboratory investigations on the long COVID cohort, which was put together by Gail Matthews and David Darley at St Vincent's. Stu Turville and his team listed there, along with Bill's group, really championed all the neutralized antibody studies and the viral isolation. None of this could have been done without the excellent blood banking done at the St Vincent Centre for Applied Medical Research, led by Kate Merlin. We've got a great collaboration with CSL Behring, the VIM cohort led by Tony Cunningham, and the funding is from those organisations there. Thanks very much.
Tony, amazing stuff. Questions, please.
Sorry. Would you like to speculate on whether your studies might give an indication of what might happen in China, in the sense that it's a predominantly I know. It's a BA5 driven wave. You know, what is the risk then perhaps of one of the BA2 variants coming in, like the XBB 1.5 and that sort of thing? You know, what might happen there?
So as you know, and I didn't go through that phylogenetic tree, but some of those current variants are, you know, through recombination of variants across those families. But it still doesn't give you a big jump in either immunoevasion or TMPRSS2 usage or infectivity. So I guess from that, it seems with the list of mutations that the virus can tolerate at the moment, it's resorting it in as many ways as it can while maintaining infectivity, but it's not actually then another quantum leap in its immunoevasive properties. So I'd speculate that maybe not too much. Again, this is me being optimistic, which you know is very atypical.
And just very quickly before I give it to you, Phil, what work has been done on long COVID immune markers and so on in kids, given the previous talk? Yeah.
I'm less across that, I have to say. So I'm going to pass that question, sorry.
But there is a question here.
I just wanted to ask Tony Philbritton from Sydney too, whether you've looked at reactivation of beta gamma herpes viruses in convalescence, and what do you think of that if you haven't looked at it in your cohort?
We haven't looked at it. I'm aware of the work. There does seem to be some correlation there, but I think more work needs to be done in that area.
Tony,
and you know, with all that activation going on, it's not that surprising that you would have some increase in other viral activation. So I guess is it cause or effect? Is it the chicken or the egg, right? So is it there? Are they being reactivated because of the immune activation, or is their reactivation causing the immune activation? And I guess from natural history studies, that's going to be really hard to dissect.
Tony, the markers might predict the symptoms in long COVID, but do they explain the symptoms?
Well, I think if, type 1 interferons, we know from the therapeutic use of interferon alpha and beta that they do have neuropsychological side effects. When we were treating hepatitis B and C with interferons, one of the major complications was depression and neurocognitive disturbances and fatigue. And so, it is plausible. Obviously, they were given at much higher doses than those levels that we were seeing, but it is plausible that those cytokines could be causing those sorts of symptoms.
Microphone's here. You're not looking this side at all, Lou.
Oh, sorry.
Tony, I'm just wondering, there was a lot of crossover between the levels of cytokines with your control group and with your long COVID group. Do the outliers correlate with more severe long COVID symptoms?
So not just when you count symptoms, and that's why we're having a close look at that with those validated scales. So that's what we're looking at the moment, and we're trying to get good statistical support before we dissect that too much, because we want to do it properly. So we're actually doing that analysis as we speak to try and look at severity as measured by accepted scales that were done at the time, prospectively against the level of cytokine. That's one of the things we're looking at the moment. Unfortunately, I don't have that data ready for this presentation.
John, I've just been given that signal. So, Tony, thank you. Fabulous talk. Really important work. I'd like to introduce the final speaker, and we started this evening with the Deputy Director of the WHO Collaborating Centre, and we're going to complete the evening with the Director. And so Kant is going to talk to us about lessons learned from